My dear students, in continuation to biotechnology and its applications, we are studying the principles of genetic engineering. The principles of genetic engineering. What genetic engineering involves. How do we construct an artificially recombinant DNA molecule? How do we construct an artificial recombinant DNA molecule? A recombinant DNA molecule is a DNA in which a desired segment of DNA taken from another organism is put into combination for its introduction into the host. This is the recombinant DNA. The construction of the first recombinant DNA emerged from the possibility of linking a gene encoding antibiotic resistance. The scientists found that there is a gene for the antibiotic resistance that codes for the antibiotic resistance with a native plasmid autonomously replicating circular extrachromosomal DNA. The plasmids are found in the bacterial cells, which are extranuclear DNA segments. The first recombinant DNA emerged from the possibility of linking a gene encoding antibiotic resistance with a native plasmid. This gene for the antibiotic resistance was combined with that very plasmid. The plasmid acted as a vector. So there is a formation of an recombinant DNA. This was conducted by Stanley Cohen, and one more biologist, Herbert Boyer. These two scientists conducted this experiment combining gene for antibiotic resistance taken from a bacterium, Salmonella typhimurium, combined with a plasmid, conducted by Stanley Cohen and Herbert Boyer in 1972, the first experiment in this field. By isolating the antibiotic resistance gene by cutting out a piece of DNA from a plasmid, which was responsible for conferring antibiotic resistance. This gene was obtained from the plasmid of a bacterium, Salmonella typhimurium, by the suitable molecular techniques. This gene was responsible for conferring antibiotic resistance. The cutting of DNA at specific locations became possible with the discovery of the so-called molecular scissors. How this piece of DNA is cut from a plasmid that involves scissors, but those are not physical scissors, they are the molecular scissors. Molecular scissors are more of the proteins in nature. They are molecular scissors, what we call as restriction enzymes. Restriction enzymes, they act as the molecular scissors to cut the DNA at the desired site. The cut piece of DNA was then linked with the plasmid DNA. This DNA was then pasted with the plasmid DNA. The plasmid DNA is a fragment of DNA in multiple numbers present in the bacterial cell outside the nuclear DNA. These plasmid DNAs act as vectors. These DNAs acted as vectors or vehicles that carried this fragment of desired piece of DNA. These plasmid DNAs act as vectors to transfer the piece of DNA attached to it. We know that mosquito acts as an insect vector to transfer the malarial parasite in the human body. Whenever we study the malaria disease, we say that this disease is contagious, it is infective and transmitting from one person to another person, but the mode of transmission, that is the mosquito, which carries the eggs, which carries the larvae from one organism to the another organism and acts as a vector. So this plasmid DNA acts as a vector to transmit this desired piece of DNA into the host cell. In the same way, a plasmid can be used as a vector to deliver an alien piece of DNA into the host organism. That alien piece of DNA, alien means a strange piece of DNA that is spaced with this plasmid DNA, then incorporated into a host cell, desired host cell. The linking of antibiotic resistance gene, which acts on cut DNA molecules. The linking of antibiotic resistance gene with the plasmid vector became possible with the enzyme DNA ligase. The DNA is cut by the molecular scissors. They are restriction enzymes. Then the pasting or ligating is done by another enzyme. That enzyme is called a DNA ligase. As we can see in the replication of DNA, the DNA is first of all cut naturally. The enzymes are already present there. Then DNA polymerase and DNA ligase help to bind the DNA again after the replication. So this DNA ligase acts as a ligating factor or combining factor for the combination of this alien piece of DNA into the plasmid DNA. This makes a new combination of circular autonomously replicating DNA created in vitro and is known as recombinant DNA. Whenever this alien piece of DNA combines with the plasmid DNA, it forms a recombinant DNA. When this DNA is transferred into replicate using the new host's DNA polymerase enzyme. When this DNA is transferred into Escherichia coli, E. coli bacteria. When this DNA is transferred into E. coli bacteria, closely related to Salmonella. This is also related to the Salmonella. That Salmonella is responsible for causing the typhoid. They are closely related. It could replicate using the new host's DNA polymerase enzyme. Automatically, the DNA can replicate as its natural capacity and make multiple copies. The ability to multiply copies of antibiotic resistance gene in E. coli. This technique is called as cloning of the antibiotic resistance gene in E. coli. Hence, to infer that there are three basic steps in genetically modifying an organism. The first step is identification of DNA with desired genes. The second important step is introduction of the identified DNA into the host. The third step is maintenance of introduced DNA in the host and transfer of the DNA to its progeny. This is what we infer or have the significance from the principles of genetic engineering. That's all for today.